Hello and a warm welcome to everyone to our today's webinar on designing phase 1 trials for biosimilars, unique challenges and strategies. I am your host Bhavika Maheshwari and I represent Veeda Life Sciences. As an integrated, tech-driven global CRO, Veeda Life Sciences is offering a comprehensive portfolio of preclinical, clinical, and bioanalytical services for our global clientele to support their innovator, biosimilar, and generic drug development programs. With an extensive KOLs situated across 26 geographies, Vida Life Sciences has become a trusted drug development partner. Together, we are serving clients in pharmaceutical and biopharmaceutical industries, agrochemical and industrial chemical industries, herbals, nutraceuticals, and medical devices industries. As a facilitator for today, I would like to introduce you all to a couple of pointers before we begin the session. This session will have a presentation in the beginning, and this will be followed by a Q&A round. We have enabled ask a question feature, and you can drop in all your questions in the screen. And moving on, I would like to introduce our panelists for the day. Dr. Kiran Mathak. Dr. Kiran Mathak is the Director of Regulatory Affairs and Phase 1 Studies for Vida Life Sciences. As a distinguished leader in the Indian clinical research industry, he comes with over 4 decades of extensive experience and has a remarkable track record of conducting numerous phase 1 studies, including first in human trials and complex clinical trials. His career spans across senior positions at renowned pharmaceutical companies such as Novartis, GSK, Pfizer, and Ranbaxy. At Vida Life Sciences, Dr. Mathur leads the execution of complex phase 1 studies, and under his leadership, Vida has significantly expanded its capabilities and global presence for innovative drug development programs. Our second panelist for today, Dr. Hiren Mehta. Dr. Hiren Mehta is a seasoned professional with over 21 years of extensive experience in clinical pharmacology, medical affairs, drug development, and clinical research programs. He has been instrumental in development of 24 new drugs and nearly 50 generic products, guiding them through various regulatory submissions, including FDA, EMA, CDSCO. Currently, Dr. Mehta serves as a Vice President for Clinical Affairs at Vida, and he's leading a team of pharmacokinetics, biostatistics, medical writing, data management, and regulatory services for early and late phase clinical trials. Our third panelist for today is Dr. Jatin Vidwana. Dr. Jatin Vidwana is the head of the Clinical Research Department, and he holds a MBBS degree, and he's bringing over 15 years of clinical research experience. He has a strong foundation in clinical pharmacology, good clinical practice GCP, good laboratory practice GLP, clinical studies, and medical writing. He plays a pivotal role in advancing research excellence and is leading a team of medical doctors, clinical pharmacologists. So, without any further ado, I would like to virtually call upon Dr. Mathak to begin this session. Dr. Mathak, over to you.
Yeah, thank you very much, Bhavika, uh, for your kind introduction. And it's a pleasure to be here talking about the biosimilar products, its regulatory side, regulatory strategies, particularly related to the phase 1 studies. Uh, Dear friends, biosimilars, as such, if we look at I would like to mention a little more detailed definition, what we are seeing in the first line here, and that biosimilars is more or less similar to the generic products of the small molecules. Okay, so that is something like a synonymous for the large molecules. So biosimilars is defined. It is a product which is a biological product that is highly similar to and has no clinically meaningful differences in terms of safety or effectiveness from an existing approved reference product in the market. I'll repeat, biosimilar product is a biological product that is highly similar to and has no clinically meaningful differences in terms of safety or effectiveness from an existing reference product available in the market. But this biosimilar product should be administered with the same route of administration and the same dose and expecting the similar kind of a response as that of produced with the reference product. Now, we are also talking a lot in this definition about the reference product. So, what is a reference product? The reference product is a product which is a single biological product already approved by the regulatory authority under their guidelines, against which a proposed biosimilar or interchangeable product is compared. A reference product is approved based on, among other things, a full complement of safety and effectiveness data. Now, this reference product is the product which is introduced in the market first for that particular molecule for a particular indication. And because it is first in the market, they need to carry out all the required tests before coming into the market. And those tests would include the uh, products like the in vitro studies, in vivo studies, then also the structural fractionation, biological fractionation, toxicology studies, then submission of an IND, and then carry out phase 1, phase 3 studies, and if even required, the phase 4 studies when it comes in the market. So they need to carry out full-fledged the IND, or now which is also called as a BLA for the biological products, and that is biological license application, like NDA for the new drug application for the small molecule. It is BLA for the biological license application so far as FDA is concerned. So now we know what is biosimilar and what is the reference product. And what I, as well as my colleagues, Dr. Jatin, as well as Dr. Hiral Mehta, would cover what is mentioned in this slide, and that is global and Indian biosimilar market trends, regulatory requirements of FDA, EMA, and other important markets, why phase 1 and phase 3 trials are critical for biosimilars, what are the major differences between EMA and FDA approval pathways, what is the differentiation between the two, and the early development and phase 1 trials. And Dr. Hiren and Dr. Jatin will give in-depth idea about how the study is conducted, and it will give you a full explanation about that. And what could be the challenges in biosimilar studies or biosimilar launch, and how to mitigate those challenges so that there could be a successful launch of the product in the market as a biosimilar product. Can I have a next slide, please? Yeah, thank you. Now, when we go to the regulatory guidelines and the terminologies, there are few terminologies which are very important so far as the biosimilar products and its launches are concerned. When the biosimilar product is to be launched in the market, the company who is going to launch it will have to decide whether they would like to have the product when it comes in the market, whether it will be interchangeability with that of the reference product, or the substitutional concept as that of the reference product. And what do we mean by that? And what we mean by that is interchangeability. That means when a product is available as a reference product and then another biosimilar product is launched, this can be switched by the doctor. That means if product A is written uh, for a patient, and then the product B, but having the same molecule and is approved by the regulatory authorities as a biosimilar product, it can be switched but that can be done only by the doctor and his prescriptions. While if it is to be substituted, that will be done essentially at the pharmacy level. That means if the doctor has prescribed drug A, but if it is not available with the pharmacist can replace the product A with product B. But it's with A and B have the same molecule, the same efficacy, and the safety of the product. So the substitution can take place at the pharmacy level, and the interchangeability can take place at the doctor level. I think I hope I'm clear about it. And but that is so far as the FDA guidelines are concerned. We'll be talking when we are talking at the regulatory side, essentially of the FDA, that is US FDA, and European Medical Agency, that is European authorities. Now that what we discussed just now was that with the FDA, but for the European guidelines, what they say, we leave it to the individual European country to decide whether they should allow interchangeability of the product or the substitution of the product with that of the reference product. Now, when we are talking of interchangeability in biosimilars, I would like to give a little more explanation on interchangeable biosimilars. And this interchangeable product is a biological product that meets the requirement for a biosimilar product and is approved based on information that is sufficient to show that it can be expected to produce the same clinical result as the reference product in any given patient. So it will produce the similar effect in a patient whether you give the reference product or you give the biosimilar product. And for biological product that is administered more than once to an individual, there is not a greater safety risk or risk of reduced efficacy from eliminate for alternating or switching the between the use of the interchangeable product and its reference product. That means when you give the biosimilar product, when you interchange it, there is no difference in the effect produced by the biosimilar product to that of the reference product related to the efficacy as well as the safety of the patient. An interchangeable product can be substituted for the reference product without the intervention of the prescribing healthcare provider. That means you can interchange the product, substitute the product if it is producing the similar efficacy and safety as that of the reference product. So that is what is known as an interchangeable biosimilar product. Now, if we look at the different regulatory approaches, the in that if we see the FDA, normally the FDA will insist for the clinical trial outcomes produced by the biosimilar as that, and also conduct something like a switching trial. That is, you carry out the study, and in the patient, you then switch the product from reference product to the uh, biosimilar product and expecting the similar results. So FDA would insist for more such studies till now, but there are likely to be some changes, which I will come to it little later. Also, if we look at the European authorities, they delegate the responsibility of interchangeability or substitution decision to an individual European member. That means European country. They have to decide, their authority have to decide whether they will permit interchangeability and substitution or not. Whereas the FDA, as I mentioned, requires clinical studies, rigorous clinical studies before giving a label at the time of giving marketing authorization, whether the product can be interchangeable or substitutable or not, based on the data which is submitted by the marketing authorization holder. And therefore, the EMA demands for stakeholder confidence. Now, here, the who is the stakeholder? Stakeholder is all the patients. So the patient should get the best confidence in a biosimilar product so that they should not feel that the when he or she takes a biosimilar product in place of reference product, there will be probably compromised efficacy or the worst safety. So for them, they say that you need to carry out some studies, either a bridging study or a limited study, or the data of the reference product, or the already existing biosimilar product. One can give that data and then uh, ask for whether the product can be substituted or not, or whether product can be interchangeable or not. So, as I said, European authorities leave it to the individual country while the FDA regulates at the central level. Next slide, please. Now, here you will see that the something like the timetable, or when did the biosimilar studies started, the biosimilar launch started. In this, European authorities were far ahead of that of the US FDA. The European Union started somewhere in 2001 or so, and right from 2003 they started uh, making the guidelines for it, and the first product in the Europe was launched in as early as 2006. And there are many biosimilars which came into the market, and they have been quite successful as that of the reference product because of the rigorous approval process of the European authorities as well as that of the uh, US FDA. If we look at the European authorities, they expect the data to be available as at least 15 years of data, and uh, over 1 million patient treatment years as that of s- biosimilar products, so that the substitution can be granted. In 2022, EMA confirms that biosimilars are interchangeable with reference products. So now they have given a more liberal attitude, and they will allow the substitution of the reference product by a biosimilar product, so that the patient can get equally efficacious and safe product as that of the reference product. US FDA subsequently also followed the similar lines, and in 2023 they dropped interchangeability statement from their biosimilar guidelines. And however, they are emphasizing that whatever data is submitted for the marketing authorization of biosimilars, even for interchangeability or the substituting part of it, there has to be a real world evidence to support the safety of the switching of the drug from reference product to a biosimilar product. And in 24, they have now officially declared a proposed removal of mandatory switching studies or interchangeability. So it is now up to the marketing authorization holders to prove that the drug is as effective, the biosimilar drug is as effective and safe as that of the reference product. And if they don't have sufficient data, then only they need to go ahead for the clinical trials studies as such. So there is a lot of changes have been brought in by European authorities and by the FDA, so that the biosimilar products can come in the market as early as possible, though without compromising the safety and the efficacy of the product as such. The next slide, please. If we look at the market data, the market, if we see, as I was mentioning earlier, European Union launched the product earlier. The first product which was launched was 2006, and they have launched by now, by year 2024, 101 biosimilar products in the market. And overall biological products, I'm talking of overall biological product as a prescription product, is about 32% of the total market uh, uh, of the EMA countries. And out of that, the biosimilar share of biological market, the biological if you look at the total biological products, out of that, the biosimilar is about 35 to 38. But that means as much as one third of the volume is by the biosimilar products, so far as European authorities are concerned. And if we look at the USA, they started, the first biosimilar was approved in 2015, but in next 9 years they have approved as many as 53 biosimilar products to come in the market, and the overall biological share of the market is the 36%, out of which the 20% is the biosimilar products as such. And if we look at Canada and Australia, the Canada and Australia more or less same numbers of the product, like 58 for Canada and 55 are the number of products in Australia. And Canada started in 2009 and Australia started in 2010 by giving the first approval to biosimilar product. And again, if we look at the market, is around 33% in both, and in both about 23 to 24% is biosimilar products. So you can imagine that out of the total biological market eh, of the biological products, as many as between 24 to 33, 35%, or rather I would say 20 to 40%, depending on the country, the market share is by the biosimilar products. That shows that the earlier launch of biosimilar can bring about a good change in the market and can give a lot of benefits to the desirable patients who can take a more economical, safe, and effective product as a biosimilar product. Next slide, please. Now, if we look at the globally biosimilar approvals that have taken place, but here we have essentially restricted to the USA and Europe. As you can see, it started with 2015 in EMA, and FDA started. If you look at the last 10 years, from 2015, when the USA started, just the one product. By that time, European authorities had already approved six products because they started much earlier than the US authorities. But it is good to see there are a good number of the biological products where the biosimilar products are entering the market every year. And in last year, actually, the FDA overtook the European authorities. The FDA approved as many as 18 products, biosimilar products, for marketing authorization, as against 8 for the EMA. But on the whole, if we see, EMA till now has approved 106 biosimilar products, as opposed to 68 FDA has approved. So the EMA still is on the higher side compared to US FDA, but both are catching up, and it's good to see there are as many biosimilar products are now coming in the market, and with the relaxation of I would say the modification of the rule, the amendments in the rule, the more and more number of biosimilar products will enter into market, and we will see in next 3 to 5 years a tremendous number of biosimilar products which will be available all over the world, including India. Next slide, please. Now, there are various guidelines, various documents that needs to be submitted before one gets a marketing authorization, and each authorities have a slightly different criteria. They may not be the identical all, but there is a trend towards becoming an identical. There is a trend for becoming something like a harmonization of the regulatory authorities in allowing the launch of the biosimilar products as well as the generic small molecules. If we look at the European authorities, the animal studies are by and large is waived, but certain time, depending on the molecule and the indication for it will be used, they may ask for the animal studies, but by and large it is waived as such. The testing product is, it will be essentially very important to show the the structural fractionation and biological fractionation to that of the reference product. And if it is exactly similar to that of the reference product, then one can have a waiver of even the clinical trial that needs to be conducted for the study. Exclusivity period is 8 years data is required, and 10 years in the market. And substitution role, individual nation, as we were discussing earlier. The EMA leaves it to the individual country. There are 27 countries in the European Union, and each country can decide whether they will permit switching of the product, that is either interchangeability or the substitution of the product. It is their own way of looking at it. FDA does usually requires the animal studies, but based on the PKPD data, they can waive off the animal studies. The testing requirement, there is a detailed guidelines available, and also there is, if there is the data which is in animal studies and the fractionation data is structural and biological fractionation is identical to that of the reference product, clinical trials may not be required, and it will depend again based on the data which is submitted. And if the indication extrapolation is required, it will be carried out based on the scientific justification. And the substitution can be done, as we discussed earlier, by the pharmacist or by the prescriber himself. Similarly, for the Health Canada, also the most important part here, which is being played, is by the structural and the functional and biological fractionation, which is to be shown as that of the reference product. And again, testing requirements, the indication extrapolation, the only change in Health Canada is no substitution is approved unless proved otherwise, and you need to do a certain bridging studies before you can exactly get a permission to have a substitution or the interchangeability of the product. MHRA UK, the animal studies are usually waived. There can be testing requirements which will depend on the product specific. They did about 8 years data and 10 years market authorization and national level decision whether to allow substitution or not. Similarly, for the Australian authorities, they generally waive off the animal studies. Testing again product specific. The indication extrapolation is allowed if the compatibility is proven with the reference product. They need 5 years data, and the substitution is not automatic. You need to apply separately for the substitution and give the data which is required by the authorities for the substitution of the product. So you can see that the major authorities, most of them for biosimilar products, they do not insist for the animal studies. The important aspect is that of the testing, which is particularly related to the fractionation, that is structural fractionation, biological fractionation studies, prove it as the same as that of the reference product, and that most of the time can also give a waiver for certain clinical trials. So as you can see now that there is now more of harmonization taking place in the regulatory guidelines, and each regulatory authority wishes that the biosimilar product should come in the market as early as possible. Next slide, please. So you can have, before you one wants to launch the product, or before one wants to decides to put the product biosimilar product in the market, then it is always advisable to have discussion with the regulatory authority depending upon in which market would you like to launch the product. Now, if one wants to launch the product in USA, US authorities, that is FDA, has a different kind of the various meetings that you can ask for early development, for scientific guidance. If there are any critical issues that are coming up while developing the molecule, developing the product, and if you are ready with everything, it's always better to go for a pre-marketing authorization meeting and take the opinion of the FDA inspectors or the FDA experts, that whether whatever we have carried out, it meets with the thinking and the guidelines of the US FDA, and then only submit the BLA and wait for the approval. But certain time, if one wants to launch the product in Europe and US both together, one can have a joint meeting between the US FDA, European authorities, and the marketing authorization holder. Discuss simultaneously with both the regulatory authorities and come to a conclusion in which way, what kind of studies that needs to be carried out, what kind of studies which are common for both the authorities, so that you can save time and the cost, and you one can launch the product simultaneously in European market as well as in USA, and one can save the cost and the timeline by launching the product simultaneously and also conducting the studies in such a way that one can save the cost and the timeline. You need not repeat the studies to launch the product in other regulatory authorities. So one can have a common meeting between the two authorities and then only draw up the development plan and conduct the studies and then submit the uh, documents for the marketing authorization. Next slide, please. Well, thank you very much, listeners. And if you have any questions, I will be able to take it up after the my other two colleagues give their excellent presentation. Thank you very much for your patient hearing, and thank you, Bhavika, for your help.
Thanks, Dr. Marthak. It was a very good insight in terms of knowing what is biosimilar, what is biologics, as well as the different regulatory pathways and the approaches the marketing authorization holder can adapt to for the different kind of the regulatory requirements. So here I will be taking up for the biosimilar development part that how the biosimilar is actually whenever company or a marketing authorization company needs to develop a biosimilar, what should be the approach and how phase one clinical trials come into the picture. Bhavika, can we move to this next slide, please? As we can see, biosimilar development cycle from start to end usually requires these three steps. Step one is the analytical studies, which requires determining the drug's physiochemical profile, which means amino acid sequence, structure, and folder of the biosimilar or biologic needs to be matched by the biosimilar product. If there is any change in the same, the marketing authorization holder should justify that change along with impact on the biosimilar function as well as the stability. Usually, the analytical studies will be followed by two kind of comparative non-clinical studies. The first one is in vitro pharmacodynamic and toxicology studies, which will be usually a mandatory requirement for all the submissions, while it will be followed by in vivo animal studies as applicable, which Dr. Marthak has already explained in his previous slides. Animal studies are not always required, and different regulatories have different I'll say the different regulatories have different opinions about the animal studies as well as the different requirements. However, the last and the most important part is comparative clinical studies. That is step three. It will include two kind of trials. One is the pharmacokinetic and pharmacodynamic studies in the healthy volunteers. These are typically the phase one studies, while it will be followed by clinical efficacy trials, which will be the phase 2 and phase 3 late phase trials. Again, clinical efficacy trials are dependent on the indication of the population in which the biologics needs to be used. If a biosimilar is used for a single indication and if there is an extrapolation of an indication, again whether the efficacy trials are required or not is dependent on the various regulatory submission as well as the molecule and its indications. Can we move to next? So these are very basic terminologies of certain studies, I will say. So in vitro toxicology studies, by definition, these are the studies of potential toxic properties or of target effects in cells or tissues in a controlled laboratory setting. While the same kind of information and same kind of studies, when they are done in a relevant animal species, it falls under the in vivo toxicology studies. It will be followed by pharmacodynamic clinical studies. When we say pharmacodynamic clinical studies, they will determine the effects of a drug on a body, while pharmacokinetic clinical studies will determine concentration of that particular drug in the body. In biosimilar setup, usually pharmacodynamic and pharmacokinetic clinical studies can be combined and a single trial can serve the purpose of both pharmacodynamic and pharmacokinetic similarities. Switching studies, we have already heard a lot from Dr. Marthak. However, studies where population currently taking original biologics switches to the biosimilar to determine pre and post study effects, while the comparative efficacy studies, which are actually conducted in the patients who are being indicated for that particular drug, the clinical studies compare the efficacy of original biologic and biosimilar. Typically, it will be done by randomized clinical trials, and in practical setups, switching studies as well as the comparative efficacy studies both can be combined together. The last and not the least is comparative immunogenicity testing, where the clinical studies that compare immune response generated to both the biosimilar and the reference product in sensitive study population. With careful planning, this immunogenicity testing can be clubbed with one of the study, like either a PD and PK clinical study which is being done in healthy volunteers or in patients as required. Immunogenicity testing can be combined with that trial, or if a sponsor requires to do a comparative efficacy trials or switching study, the immunogenicity testing can be the part of that study. So for immunogenicity testing, there will be no separate study required, but it will be combined with one of the either study which can be conducted. Can we move to next, please? We have actually captured certain examples, and the example of a well-known biosimilar product adalimumab is actually here, and we have actually presented the types of the trials conducted by the different sponsors. As we can see, all the sponsors have conducted phase 1 trials as well as the phase 3 trials for different indications. It is noteworthy that Amgen has conducted a clinical efficacy trial for two indications. So instead of only one indication, they have conducted trial for both the indications. Maybe it was an extrapolation of the indication, we do not know. But still, they have done in this manner. While Sandoz and Tenacious KB kind of the sponsors, they have done it only in a single indication, like psoriasis. Can we move to next, please? And this is again continuation of the phase 1 studies which were presented in the previous slide. As we can see, almost all the trials, the duration of the days were between 60 to 70 or 72 days, the lowest being 63 and highest being 72 days. The number of volunteers or the patients required again ranges from the minimum is 108 to highest 318. All the studies have compared PK profile. The comparator, they have utilized both US and Europe, so that the single study they have actually compared their test product with the reference from Europe market as well as reference from the US market. As we can see, Sandoz also have done a study not only to prove their original product with reference but also with the pre-filled syringe administration. While Mylan, where they have done a relative bioavailability study to actually check the delivery via different routes, or I'll say different packagings, means by vial as well as by pre-filled syringes. Almost all the studies have checked the primary PK endpoints like AUC 0 to infinity, AUC 0 to time, and of course Cmax. Next. So these are the goals of phase 1 trials for the biosimilars. The first and foremost goal will be the PK similarity and key matrices as we already discussed. It is Cmax, 0 to time AUC or 0 to infinity. Pharmacodynamic similarities, which requires comparing biological activity using the sensitivity markers. And it is actually used when direct efficacy studies are not feasible or uncertainty on the PK similarity. Safety and tolerability, with the phase 1 trials, one can check whether the drug is safe and tolerable, and it can be done with a deep down access to incidence, severity, and type of adverse events, as well as evaluation of different safety parameters, including vital signs, laboratory values, ECGs, etc. As we discussed, immunogenicity assessment can be combined with that, which can detect development of anti-drug antibodies, as well as impact on PK, PD, safety, and clinical response. Correlation of anti-drug antibody level with these parameters like PK, PD, safety, and clinical response is also noteworthy and should also be presented. It also support for the clinical development strategy, which will provide data to justify extrapolation of the indications. And it will also determine the need for the phase 3 confirmatory trials, which will again be dependent on the molecule, the kind of indication, and the type of the regulatory agency the sponsor is actually intended to submit. And it can be done with the design of the clinical trials in the fashion that we have mentioned. It can be either single dose study or a multiple dose studies. Usually, the single dose study, if there are no major safety concerns, can be done in a healthy volunteers, while the multiple dose studies or a single dose study, which is having very narrow safety margins, will be done in the patients. The studies will be either open label or a blinded when possible. Usually, the biosimilar trials will be either parallel or crossover, but in our experience, it should be ideally the parallel trials because of the long washout and the long follow up that is required to be done for that particular trial. However, sponsor may choose to go for a crossover trial as required, and as I already mentioned in the previous slide, usually it will lead to a large sample size to complete a clinical trial. Next, please. And these are the key design considerations for the phase 1 biosimilar trials for a CRO. First is the study population. As I mentioned, if safety concerns are not does not necessitate the trial to be conducted in patients, healthy volunteers are preferred. However, whether it is healthy volunteers or patients, careful subject selection to have very low variability and minimal confounding factors to enhance the sensitivity is an important factor. Study design we have already discussed, randomized double-blind crossover or parallel with long half-lives, and standardized conditions like fasting, washout periods, etc. needs to be taken care to minimize the variability. Endpoints primary will be the PK parameters, which will be Cmax and AUC, as well as the different pharmacodynamic parameters based on the molecule mechanism of action. The secondary endpoints will be safety and tolerability, and of course, occasionally immunogenicity. The last one is the important aspect because it is challenges in the multi-regional or country clinical trials. Certain European agencies expect that the population in the clinical trial is mixed population and it is the population is actually present across the regions, across different countries. They do not want the study only in the Caucasians or only in the Asians, so they may ask for that variability or they may expect the variability in the population. In these kind of studies, role of CRO becomes most important or more important because it comes with the challenges of logistics of harmonizing protocols, ensuring consistent sample handling methods, and managing population heterogeneity. However, an experienced CRO, of course, like Veeda, we are having enough capabilities to manage this and mitigate all these risks to make sure that the trial is conducted as per the requirements without much of the variability. Next, please. Why an experienced CRO is a requirement for a biosimilar trial? I'll say the experienced CRO can bring on the table the various advantages. The CRO can work optimized study design, which includes adaptive trial designs as well as two-stage innovative approaches. The two-stage innovative approach, I will like to have one more minute on that. It is a trial where Number of volunteers will be dosed, their PK and PD parameters will be analyzed, and based on the results of those, the further course of the trial in terms of whether to continue the trial, whether to increase the more number of the volunteers or the patients, or to include more number of patients and healthy volunteers in the trials will be decided, and accordingly the study can be completed. However, all this needs to be very predefined at the start of the study in the protocol, and these plans needs to be approved from the regulatory agencies beforehand. Of course, the express CRO will be having a state-of-the-art bioanalytical capabilities with different kind of the LCMS, MS, ICP, OES, or capabilities of ligand binding assays. Having single methods and low run time can also help, or I'll say, expedite the project timelines up to a large extent. Efficient site management capabilities to control variabilities and expertise in global regulatory compliance, as well as the multi-regional country trials. Of course, a speed of a trial will be defined by recruitment of the patients as well as the LD volunteers. Diverse and rapid patient pool as well as the volunteer pool is also a considerable factor to decide the CRO as well as to decide at which site the study should be conducted. Last and the most important part is the quality framework. An experienced CRO brings on the table robust quality and compliance framework, which will make sure that all the GLP certified and GCP compliant facilities are working on the studies, as well as all the activities done at the CRO are within the scope of the, or I'll say, are within the compliance of the GCP and GLP. Next, please. These are the examples of biosimilars and challenges for a CRO. These are actual examples of the studies being conducted at our sites. First is the omalizumab, and as you can see, for all the studies, the submission was both Europe and US. Omalizumab, we required more than 200 to 300 subjects to be enrolled. The recruitment required the healthy volunteers who are non-smoker and their IgE levels less than 100 international unit per ml. It was having lung ambulatory visits. We, to complete the recruitment in timely manner, we use multiple clinics, two different sites across the region to complete the recruitment in time, and our rigorous follow-up with the subjects led to a very minimal dropout. Vidolizumab is again an interesting case. When we went to regulatory agency of India, they asked us to conduct the study in a hospital-based setup. So volunteers were identified and screened at Veeda. However, all the clinical activities, including admission, dosing, as well as sample collection, sample management, most importantly, drug management, pharmacy management, everything was done at the hospital site by CRO team. Pilgrastim again is one more example that has been conducted. However, the take-home message is selection of right kind of volunteers, having execution capabilities of sample handling as well as the IMP handling, and rigorous follow-up with the volunteers to complete the long ambulatory visits are the key important factors to complete any clinical trial or any biosimilar clinical trial, I'll say. Can we move to next? This is again the same different examples, but as I have already told, the same take home message is here: selection of the right kind of volunteers, extensive training to pharmacy and nursing staff to handle the IMP right from the receipt till we dose the volunteer, and at the last, maintaining the accountability and retention of the samples. Everything needs a good experience by a team. Thanks. Next, please. So, with here, I will stop my talk. As already mentioned by Bhavika, we will take the question and answers at the end of the session. it will be this will be followed by outcomes from the different phase 1 clinical biosimilar trials, and it will be taken by my colleague Dr. Hiren Mehta. Thank you.
Thank you, Dr. Marthak. Thank you, Dr. Jatin, for an excellent overview of different regulatory frameworks and how to successfully plan a study. The next step for a successful submission of the overall dossier of a biosimilar is what will be the outcomes from a phase 1 biosimilar trial and how to interpret, how to submit them to the agency. I will walk you through some of the slides. For example, the first slides on the left hand side, you will see a PK graph, which actually shows a PK similarity between the two products, the test biosimilar as well as the reference biological product. This is an example from adalimumab. You can see that the red line is for the reference biological product and the black line is for the test biosimilar product, almost superimposable with the reference product at all the time points. This we can definitely say that the area under the curve as well as the peak concentration are almost similar between the two products. So the study actually proves a similar PK profile. The agency from this phase 1 study also expect the effect of anti-drug antibody presence and how that ADA positivity affect the test and the reference product. Do ADA affect them differently or in a similar fashion? On the right hand side, you will see two different plots. The first on the top is actually a test product plot for the subjects in the black line showing the concentration levels who did not report any anti-drug antibody versus the red line with the subjects who actually reported positive immunogenic response. So there is a difference between the two curve. Normally, subjects showing the positive anti-drug antibody tend to show lower plasma levels compared to the subjects with a negative response. Similar is the case on the plot below, that is for a reference product. The subjects who reported positive anti-drug antibodies actually presented with little lower plasma concentration at almost all the time points. So in this slide, we can see that apart from a PK similarity on the left hand side, even the effect of the anti-drug antibody are almost similar in test and reference product with a slight difference near the peak level, but otherwise the area under the curve reduced in subjects who were reported the positive anti-drug antibody. Next slide, please. Apart from the PK similarity and the effect of anti-drug antibody, we also have to present to the agency what is the percentage ADA positive rate at each visit. In this slide on the left hand side, you will see two plots, the box plots, the top one and the bottom one. The top one actually shows us the percentage ADA positive response rate at different visits. In this slide, there are three visits, day 14, day 42, and end of the study, day 70. If you see the three bars, all of them, the black ones are actually for the test biosimilar product, and the gray ones are for the original biological product. Almost comparable in most of the visits. Initial two visits, the test product actually showed more percentage positive response rate, but at the end, the gray bar actually shows that the original biological product reported more number of positive rates. But in general, looks like a comparable situation if we sum up all the visits. On the lower side plot, we are presenting the percentage neutralizing potential at each visit. Here we have only selected the last two visits because at day 14 there were no neutralizing potential. In the last two visits, we can see that almost comparable between the test and the reference. So in a nutshell, compared to the previous slides and this slide, there was a PK similarity between test and reference. The effect of anti-drug antibody response was kind of similar between test and reference in terms of the plasma concentration and or overall exposure of the drug in the body. Also, Comparing each visit, the percentage ADA positive rate as well as neutralizing potential was similar between the two products as well. On the right hand side, we are seeing a comparison of adverse event profile or safety profile between the two treatments, the test as well as the reference biological product. The total number of adverse events were kind of similar between the two, close to 50%. The category of adverse events are also pretty much similar for all kind of adverse events. So combining these two slides, we can interpret that this study was actually a successful submission with a PK similarity, similarity in adverse event rates and overall safety profile, as well as the immunogenic response. This is typically what an agency expects from a successful biosimilar trial. There may be a component of a biomarker if needed. Because this submission was pretty clean, a biomarker was not required. Otherwise, an applicant can choose to include a biomarker element in the study as well and show a similarity with the reference biological product. Next slide, please. So normally after, the way Dr. Marthak and Dr. Jatin explain how we should plan and how we should conduct the study and we submit the dosage to the agency, there has been some recent changes in the agency's, review process as well as their thought process. And these are some recent regulatory feedbacks from our different sponsors across the industry. And the feedbacks are divided into two parts. The first one on the top is about phase one, and the bottom one is about phase three. In phase one, normally the agency delayed or rejected the whole application because of an unsuccessful PK or PD comparability. So PK and PD comparison is must to the extent they should look similar. The agency also sent some queries on inappropriate crossover models where the washout was not sufficient. There was a carryover from the previous effect, the previous treatment effect, or the study overall after completion did not meet the desired power, which was assumed during the protocol development. There were also some queries for the assay sensitivity because we are dealing with very low levels here for the large molecules. So the method should of course be sensitive enough. And also some of the agencies send a query for selection of wrong PK endpoint. So some applicants missed partial AUC, some applicants chose a different partial AUC. So we have to be careful in designing the protocol itself. Some queries were related to the inappropriate PK-PD correlations. For example, PK was collected at different time points and the biomarker or a PD levels were collected at a different time point. So basically there was no PK-PD correlation possible because of different time points. Or in some study, the PK similarity was proven. However, the main biomarker or a PD endpoint was failed and the agency send the query to prove the PD biomarker similarity as well. For phase 3 trial, normally the agency look for a sufficient immunogenicity data that I saw in my previous slide. That was from a phase 1 study. But in phase 3, they look for a large scale immunogenicity analysis, mostly after certain doses. They want to check the similarity of immunogenic response between the two treatment of test biosimilar as well as the reference biological, and they should be kind of similar. In some of the studies, there were lack of proper antidrug antibody assessment with the neutralizing potential. Some applicants do go for ADA assessment but forget the neutralizing potential, or they do not compare the neutralizing potential between the test and the reference. That also results in a query. Some applications go for a primary indication as well as extrapolation of indication, but they fail to provide a justification that why an extrapolation to a different indication should be justifiable without the need of a clinical trial. So agency asks for a proper justification if you want to extend the extrapolation, if you want to extend the indication as well. Some sponsors fail to select a proper reference product in a multi-country trial or reference treatment regime because of difference in practice at the clinic level, and they end up having a query of why the product was not administered similarly across all the treatment arms. So these are basically a summary of what kind of query an applicant can receive for a biosimilar submission, and that's why use of an experienced CRO is must to avoid this kind of situation at the end. Now let us focus a little bit more on the Indian CROs in the biosimilar development. There has been a lot of noise and a lot of promises from the Indian biosimilar development, including some of these CROs. So we'll walk you through the recent developments in Indian CRO industry, especially for a biosimilar. So in India, the regulatory pathways are very clear. Either it may be a bioequivalence or bioavailability study or a clinical trial phase 3 study. There are two different regulatory pathways. For a BAB study, normally the approval timelines are between 50 to 60 days. And for a clinical trial, the normal timelines for an approval is around 4 to 5 months. In most of the cases, this goes to an SEC meeting where the expert from the industry or academia discuss the submission and approve or ask for an additional data. Normally, for most of the biosimilar product which are injectable, the agency expect a preclinical or toxicology study in one or two species based on the guideline requirements. Now in this slide, I was planning to show you the number of biosimilar clinical trials in India in last 5 years by both sponsor as well as CRO. If you see in 2020, the number of trials were kind of less, around 25 for the whole year, combining sponsor-led or a CRO conducted clinical trials. And there has been consistent increase in number of biosimilar trials in last 5 years. In 2021, it reached up to 32. In 22, more than 65 trials. In 2023, more than 70 plus trials. And last year, by end of the year, almost 84 trials in biosimilar space were conducted in India, either by a sponsor or a CRO. However, in general, in last many years, CRO is actually leading the way in terms of total number of trials. In last 5 years, more than 150 trials were conducted by CRO and more than 125 trials were conducted by sponsors themselves. So this proves that there is a capability, there is enough number of clinical trial site, and the market is going upwards in next 5 years as well. In this slide, I was planning to walk you through the future trends in a phase 1 biosimilar trials with a lot of technology in place, and everybody is trying to utilize or leverage the existing technology available in the market. I could I could figure out that the future of biosimilar is in combination with real clinical conduct plus AI and machine learning, especially in predictive modeling when the FDA has now proposed to eliminate the switchability trial. Sponsor will now go for a predictive modeling for an indication or extended indication with a PK/PD correlation as well as the exposure response analysis using the AI/ML modeling techniques. And currently Pumas as well as Certara both offer these services, and this can help you either minimize the trial cost or reduce the timelines as well as help you get a complete waiver for any clinical trial after the phase 1 PK/PD study. In the FDA and Europe as well, the real world evidence will actually help beyond the clinical trials to get real patient data with a parallel use to prove to the agency that the product actually works well in the clinic and it mimics the actual clinical trial and supports the earlier outcome from a phase 1 study. So real world evidence is also going to play a major role. There has been shift in the regulatory pathways and the India's role has been expanding. However, EMA recently has been asking for certain number of Caucasian population in the trial if the dossier was supposed to be submitted to the any of the EMA country. Normally, they ask for this data to check the impact of ethnic variation in the cytochrome enzyme as well as other variable factors that can contribute to the overall efficacy of the product. That will ultimately go to a decentralized clinical trial pathway in phase 3 or phase 1, where there will be multi-country sites, there will be a mix of population and remote monitoring required, and different data analysis techniques will also be needed to see the difference in the ethnic variation at the end of the study. Dr. Jatin already covered the risk mitigation approach because most of the biosimilar phase 1 or phase 3 trials are very large sample size, very long duration and a costly affair. So rather than going with a two-way crossover or a parallel study, once we start the study and only wait for the results at the end of the study, will actually can go wrong in some cases. That's why we suggest two-stage design approach where we will see the power and the trend of a bioequivalence summary in the interim after we complete about 40-50% of recruitment. In recent development, some of the sponsors have been exploring the possibility of futility analysis middle of the trial, which can help them go or no go decision based on the interim results. So that can save you a lot of time, cost, as well as the timeline, and you actually know where the study is going and in which direction you should move forward. So overall, based on the previous slides, the major takeaways from the whole discussion is that the biosimilar approval pathways are actually constantly emerging, even in a regulated market. The recent change in mindset of FDA for waiving the switchability trial and the EMA asking for real-world evidence data at the same time approving the dossier is actually a continuous improvement in their approval pathways help the patients to get the effective medications in a smaller duration. Someone has to actually adopt the unique study designs, alternate approaches, as I said earlier, the two-stage design as well as the futility analysis, or combined trials for phase 1 and phase 3 to minimize the overall duration, the timelines and the cost as well as the efficiency of the study. Now parallel development is possible for both EMA and FDA in the same protocol. An agency recommends and encourages the applicants to consult both the agency at the same time. The futuristic trial, especially when EMA and FDA expecting the population to be of a mixed nature from a particular country population as well. So the future trial will be across many countries and many populations, and we need to design the protocol in such a way. India can of course become a global hub for biosimilar phase 1 trial as we have seen in one of the slides. Number of biosimilar trials are constantly increasing and that too for a regulated market as well. And Veeda of course significantly contributes to the biosimilar development as a whole and have done a lot of biosimilar trials in recent past. I think with this, I will pause and hand over the webinar to Bhavika for question and answer session. Thank you so much for your patience and being a wonderful audience.
Thank you to our speakers for a great presentation. I'll just quickly look at the questions. Okay, so our first question is, how is immunogenicity assessed during phase 1 trials of biosimilars, and why is it important? Dr. Mathur, can you please answer this question?
Thank you, Bhavika, and thank you for a very important question because the immunogenicity plays a very important role in any biosimilar product. And immunogenicity, as we all know, is assessed by measuring the anti-drug antibody testing, and also the neutralizing antibodies, that is NABs, in blood samples which are collected before dosing the patient and after dosing the subject. So far as phase one is concerned, the because the dose is sometimes is on the lower side, the duration is not as much as one would like to have for phase three clinical trial. And but still, it is important to have the analysis of the ADA, that is anti-drug antibodies, and neutralizing antibodies, so that one can come to know at an early stage what is happening with both the ADA as well as NABs, and one can make out about the immunogenicity effect of the drug. So it's very important to do that even in the phase one study, and it can therefore help in also come to know about what kind of risks are involved when one wants to go for the phase three studies, and how one would mitigate those risks when conducting the phase three study in a larger patient population, and also invariably for a prolonged duration of treatment. And therefore one can decide about the how the monitoring of the study needs to be carried out. So it helps in even designing the protocol, particularly for phase three. Yeah. Thank you.
Thank you so much, sir. My second question is, what does what role does statistical analysis play in establishing biosimilarity during phase 1 trials? Dr. Hiren, can you please answer that question?
Yeah, thank you, Bhavika. I think I can take this question. So, in a biosimilar, especially in phase one and phase three trials, the statistical planning is very important from the beginning of the protocol or study design, especially for the power of the study, the estimated variability between the test and the reference product. The 90% confidence interval, of course, is the key requirement for showing a similarity of PK endpoints, which is Cmax and AUC. Normally, they expect 90% CI between 80 to 125 for a parallel as well as a crossover design. But especially when we go for an adaptive trial design, either futility analysis in the interim or two-stage approach, the statistical analysis actually contributes in a bigger way compared to a routine study. If I talk about the two-stage approach where we check the power as well as the bioequivalence limits in the interim of the trial, use of a proper method and alpha inflation or alpha penalty is very important, and that is what agency expects. We normally use, I personally recommend Portwin method C for two-stage approach, which is most promising method for parallel and crossover both, which also help us determining the alpha penalty as well as the move forward for additional number of patients. So in general, statistical analysis during the design as well as in the interim analysis, as well as at the end of the study when we are about to submit the report, helps a lot in overall study plan and reporting. I think I answered that question. Thank you.
Thank you, Dr. Hiren. My next question is, can a single phase 1 PKPD study be designed to support biosimilarity submissions to both FDA and EMA using reference products for both regions? Dr. Jatin, can you please answer this question?
Yes, please. Yes, it is quite possible that a sponsor can do a single protocol with PKPD study in which a test reference is compared with US reference as well as a Europe authorized product. However, it will require a careful planning because both the regulatory while accepts a study to be done, a single study done for a single test product, including both the arms, they have certain expectations which needs to be made. They may require a different careful consideration of the BE criteria requirements. It will also require that the protocol mentions the different kind of comparisons in terms of the taste comparison with the reference of Europe, taste comparison with the reference of US, or optionally both the reference comparisons between both the reference products. I'll say this kind of trials will actually help a biosimilar development to a large extent, especially when both the agencies are currently actually accepting and adapting the totality of the evidence approach, because it will streamline the development, it will reduce the cost and timeline involved in the particular trials. So yes, it is quite possible, feasible, however, with careful planning and careful justifications embedded in the protocol. Thanks.
Thank you, Dr. Jatin. I would like to take a pause over here. I understand that this session has kept us all very engaged, but due to certain time constraints, we won't be able to take any more questions. But please be rest assured, we will be emailing answers to your questions along with the link of this recorded webinar. Lastly, on behalf of Veeda, I would like to sincerely thank everyone for being here, taking out the time and keeping us all engaged in this webinar. Thank you to our panelists, Dr. Kiran, Dr. Hiren, Dr. Jatin, for being wonderful presenters and giving us some great insights. Thank you to our audience once again for tuning in. We at Vida Life Sciences earnestly look forward to hosting you all again. Till then, take care, stay safe. Thank you.
Thank you.
Thank you.
Thank you all.
Thank you. Bye.